Today our topic is animal cell. Animal cell has various organelles and these organelles are performing their specific functions in the animal cell. We'll start with the nucleus. Nucleus is the central organelle of the animal cell and nucleus has a dense structure called nucleolus inside it, and nucleolus forms ribosome. Nucleolus forms ribosome. This is the function of the nucleolus which is present in the center of the nucleus. Then nuclear membrane or nuclear envelope has certain nuclear pores, and these nuclear pores enable the molecules to go out or the chemicals to go out or get inside the animal cell as the need may be. Then the material which is present inside the nucleus is also called nucleoplasm, and nucleoplasm has its own function in the animal cell. Animal cell has other structures which are performing their specific functions. For example, the jelly-like substance present in the animal cell which is present throughout the cell, and its function is to provide a site for the various chemical reactions to take place, and it is called cytoplasm. Then there are vacuoles, which are the spherical structures present in the animal cell, and the function of the vacuole is to, in unicellular organism, they are divided into contractile vacuoles, removing excess of water from the body, and food vacuoles, digesting the food material. Similarly, in the plant cell, they are functioning, that is, in a way that many small vacuoles are present, and whenever the plant cell is about to get mature, then they absorb water, fuse, and form a central vacuole in the plant cell after its maturation. Then there are the structures which are called centrosomes. They are also present in the animal cell, and the two centrioles, they collectively form a centrosome, and the function of the centrosome is the spindle formation during the cell division. Moreover, in certain cases, they are involved in the cilia and flagella formation. Then there are microtubules. These are the tubules which are present throughout the cell, and they are made up of a protein called tubulin protein, and they are enabling the animal cell to hold its shape. Similarly, there are also microfilaments, and microfilaments are also present throughout the cell, and their function is to change the shape of the cell with the help of a protein called pectin protein, because cell during its activities sometimes needs to change its shape, and this process is done, or this function is done by the actin protein. There are lysosomes which have the digestive enzymes, so their function is to digest the food material inside the cell. Similarly, mitochondria are also structures which are present in the animal cell, and mitochondria are also called powerhouse of the cell because their function is energy production. Mitochondria have oxidative enzymes which speed up the reaction by which energy is produced inside the cell. So, mitochondria are called the powerhouse of the cell. Golgi apparatus, also called Golgi complex, is also another important organelle of the animal cell. It is also present in plant cell, and the Golgi apparatus forms the Golgi vesicles. These vesicles pack the foreign particles coming from the various places and then pack them and also send them to the places where they are needed. Golgi complex also performs another specific function, that is, it decides that which protein will come inside the cell and which protein will go out of the cell. Why? Because animal cell have outermost covering which gives it a specific shape called cell membrane. And cell membrane has certain pores in it which are very small microscopic pores. That's why with the help of these pores it allows only certain or selective molecules to come inside and prevents the other from going. So that's why cell membrane is also called selectively permeable membrane. Selectively permeable membrane because it allows only selective particles to go inside and also to go out. Similarly, we have microvilli. Microvilli are the structures which are present inside the animal cell, and these microvilli enable the animal cell to increase its surface area. So microvilli increase the surface area of the animal cell. Then there are other specific structures as we are knowing that there are centrosomes present, and these centrosomes are, I just told you, that is previously I've told you that these are present, these are used for the formation of the cilia and flagella. Moreover, during the cell division they form spindle fibers, and then they also form microfilaments and microtubules. This function is also done by centrosomes. That is the formation of the microfilaments and microtubules. I have just explained the function of the microfilaments and microtubules here. That is, uh, microfilaments are having actin proteins and they change the shape. Microtubules are having the tubulin protein and they hold the shape. Similarly, there are three ribosomes present inside the animal cell and their function is protein synthesis. Then endoplasmic reticulum, which is of two types. First is the rough endoplasmic reticulum, which appears rough due to the attachment of the ribosomes in it. We can see that ribosomes are, I can also show you by forming the structure of endoplasmic reticular having, these are the ribosomes which are attached on its surface. So its function is protein synthesis. Another type of the ribosomes, smooth ribosomes, smooth endoplasmic reticulum, sorry, is smooth endoplasmic reticulum is without ribosomes, and its function is cell secretion, transport of the calcium ions, and also detoxification. So these specific functions are performed by the smooth endoplasmic reticulum. And this smooth endoplasmic reticulum looks smooth because it does not have ribosomes attached to it. So this is the overall structure of the animal cell, and I'm hopeful that you must have understood the functions of the animal cell. Once more briefing, I can say that to sum up, that nucleus is the brain or control center of the cell, and it controls the entire cellular functions, and cytoplasm is the jelly-like substance which provide a site for the various chemical reactions to take place, and microvilli are increasing the surface area. Centrosomes are used for the spindle fiber formation, cilia and flagella formation, and microtubules enable them to hold the shape having tubulin protein, and the microfilaments change the shape by actin protein. Lysosomes are digesting the food particles. Vacuoles are removing extra waters in unicellular organisms, and also they are called contractile vacuoles. And the food vacuoles digest the food particles. In the similar way, mitochondria are used to produce energy for the performance of the various activities, and Golgi complex are deciding the protein particles which protein particles will remain inside the cell and which protein particle will go out of the cell. Similarly, ribosomes, endoplasmic reticulum are of two types, that is rough endoplasmic reticulum are forming protein, and the smooth endoplasmic reticulum are involved in the cell secretion, transport of the calcium ions, and detoxification of the various particles. I hope you must have understood it.